Good afternoon, everyone, and a warm welcome to our webinar, Navigating Downstream Bioprocessing for Biosimilar Development. I am Apurva Mahajan, Communications Manager at Biospectrum Asia and your moderator for today's session. Today's webinar will begin with an overview of biosimilars, highlighting the key challenges faced by developers in this field, particularly focusing on aspects like regulatory compliance. Following that, we'll introduce Praesto AP+80, a high-capacity protein A resin specifically designed to support large-scale biosimilar development. Finally, we'll dive into a cost comparison study offering an in-depth analysis of the features and costs of Praesto AP+80 in comparison with competitor resins. So without further ado, let's welcome our speakers for today. First, we have Mads Ramberg, the Senior Director of Product Management at Ecolab. Mads leads the development of products and go-to-market strategies for Ecolab's Purolite affinity resins. With 28 years of experience at Cytiva, where he held various roles in R&D, product management, and commercial functions, Mads brings a wealth of expertise to our discussion today. Joining Mads is Jo Jo, also known as Jake, who is a global field application scientist at Ecolab. Jake specializes in supporting customers across South Korea. He holds a degree in cancer biology from the Ulsan National Institute of Science and Technology in Korea, along with a master's degree in science and technology. Before his current role at Ecolab, Jake spent 10 years at Cytiva as an application specialist, helping to solve complex challenges in this field. Mads and Jake, thank you for joining us today. The virtual stage is all yours.
Thank you. Before we dive into today's topic, I wanted to take a few minutes to introduce Ecolab, review our values, and explain how bioprocessing and purification technologies fits into the larger mission of Ecolab. For 100 years, Ecolab has worked to improve health of people, the planet, and businesses around the world. At Ecolab, we aim to bring science-based solutions, data-driven insights, and world-class services to advance food safety, help maintaining clean and safe environments, and optimizing water and energy use. Today, we're going to discuss the solutions we're bringing to the healthcare and biopharmaceutical industries, particularly into the development of biopharmaceuticals. As part of Ecolab Life Sciences, we remain firmly committed to downstream bioprocessing with a clear focus on modern resins to meet current and future purification demands in clinical development as well as commercial manufacturing of biopharmaceuticals. We are continuing to expand our manufacturing capabilities, both scale out at current sites, but also addressing capacity in new sites, all in all with aim to maintain a very high level of supply security. Significant investments in R&D are making sure we put the best products on the marketplace for current and new modalities. To support our customers, whether operating at a small scale or building to commercial scale, we offer on-site and virtual technical support services. This service also involves a bioprocess lab where we can conduct studies on our customers' behalf, as well as hands-on support in every region across the globe through our field application specialist team. Our industry partnerships together with Repligen ensure we stay at the edge of new affinity ligand developments through advanced library screening technologies. We also offer insightful regulatory expertise to support our customers during their developments. Finally, our quality assurance team makes sure we have a systematic and up-to-date approach to secure we meet the expected quality standards of the industry. As patents of originated drugs have expired, the biosimilar market has experienced a significant growth in the recent years, a growth that is estimated to continue over many years to come. This has led to more affordable medicines for patients all over the world. One important enabler is that regulatory bodies have issued specific guidelines for developing biosimilars, speeding up the time to market from a 10 to 15 years time span for an originator drug down to 6 to 8 years for a biosimilar drug. Physicians have also embraced the biosimilars as safe medicines, which is increasing the uptake amongst the patient population. Improved and more cost-effective manufacturing processes are important drivers to reduce the overall cost of goods and making the medicine more affordable. This includes high productivity resins, as well as an increasing development of expression levels from modern cell lines. An overall increase in competition drives the need for cost-effective manufacturing solutions even further, though. The development of biosimilars relies upon several factors, but in essence follow the same path as for the originator drug. Enhancing process efficiency by applying effective tools like design of experiments, as well as a careful selection of the capture protein A resin that is predictable, close to the originator process, and scalable. Ensuring product quality, securing biosimilarity between the originator drug and its biosimilar to meet regulatory compliance standards. This is where most of the work needs to be done, actually. Consider cost of raw materials such as resins at an early stage to make sure the final commercial scale has resulting in a competitive cost of goods structure. In summary, biosimilars require bioprocessing resin that offer comparable performance, facilitate rapid time to market, and maintain process economics for affordable production. Purolite has built their affinity toolbox on a new agarose jetting technology, resulting in uniform beads which allows for more consistent flow properties and a lower carbon footprint compared to the classical emulsified technology used by leading resin providers. To cover the increasing diversity of biomolecules, we have broadened the portfolio through innovative ligand technology developments with our Repian partnership, improving selectivity as for Praesto 70CH1 targeting Fabs, increasing the elution pH as for Praesto Jetted A50 high pH, which ultimately will enhance yield for sensitive antibodies such as bispecifics. We have also addressed the needs for more cost-effective resins, most recently the launch of Praesto AP+80 to specifically target the biosimilar space, where cost of goods is of key importance to stay competitive in this field.
Thank you, Matt. Hi, I am Jake, and I am a person in charge of field application scientist. Let's start the introducing session of Presto AP+80. It was announced this year. Historically, emulsified APC plus resin was existed, and it was jetted version of APC plus with fortifying their function too. Let's look at the more detailed information. Presto AP+80 is engineered to support large-scale purification processes necessary for the manufacturing of biosimilar medicines. It offers comparable performance and improved process economics. As it has an 80-micron bit size, it can make maximized flow rate and throughput. As it is designed for biosimilar development, it supports large-scale protein purification and manufacturing site. About protein A resin, you know, alkaline stability is directly impact on the economical burdens. AP plus 80 has enough alkaline stability and DBC. We will check it out later. Ecolab has very powerful worldwide regulation team independently. We are supporting seamless resin transition with performance equivalence and reduced cost of goods from clinical phase to approved. Testing was performed in 0.66 ID with 10 cm packing height column using a 5 mg per ml human IgG applied sample in PBS at pH 7.2 buffer. We can see about 50 mg per ml DBC at 4 minutes residence time and about 57 mg per ml DBC at 6 minutes residence time. Similar capacity between AP plus 80 and competitor resins allows for using AP plus 80 at existing columns. To get the right result from chromatography, we need to pack the column right ways. If you are considering a packing support, don't worry. We are supporting our customers for it by global application scientists. Please contact us if you are suffering by this kind of matter. The column was exposed by zero point 0.1 normal sodium hydroxide at static condition for this page. Sample was polyclonal human IgG and DBC was measured at every 10 hours. No loss of dynamic binding capacity after 100 hours exposure with zero point 0.1 normal sodium hydroxide. And it showed greater than 90% DBC remaining. When we calculate the expected cycle number with 15 minutes contact time for CIP based on 100 hours, we can expect 400 cycles. If we think about these assumptions in a conservative way, at least 100 to 200 cycles can be expected to be an area that can be used for various processes. Let's talk about pressure. This pressure flow curve was tested on packed bed condition with a 220 cm and 25 cm bed height. Flows up to 350 cm per hour are achievable at 25 cm bed height in a 30 cm diameter columns within the safe operational window of less than 2 bar pressure. Easy to understand about flow rate. I converted volumetric flow rate to residence time. Our residence time of 350 centimeter per hour at 25 centimeter bed height is 4.3 minutes. Residence time at 400 centimeter per hour with 20 centimeter bed height column is 3 minutes residence time. As you know, around 220 centimeter bed height and 4 to 6 minutes residence time are very familiar criteria for bioprocess. That means AP plus 80 is the resin which can be easily adapted for bioprocessing process. To give you additional information, Praesto AP plus 80 with jetted based bead requires additional compression for optimal packing performance when compared to batch emulsified-based bead. Compression factor is around 1.2 for manufacturing size columns for AP plus 80. From now, let's touch the cost comparison study on AP plus 80. Left and right side graphs show cost of pre-packed column with Ecolab's competitor. First, left graph is about 14 cent ID with 20 centimeter packing height column. We can save the cost about 32% regarding to competitors. Right graph is about 45 centimeter ID with 20 centimeter packing height column and 28% cost saving is achieved. The prices of prepackaged columns you can see on the graphs are including resin, column hardware, and packing fee. A company which should use prepacked column for their process, the prepacked column make an economical issue by their price, but Ecolab have been offering it with more friendly and gentle mind, as you can see with highest quality. If you need to consider both the prepacked column and the sustainable types, please contact us. We can help to make a conclusion what type of column is better to you. Until now, we are checking AP plus 80 with the benefits we can get. Some parts are behind of price and some parts are front of it. Those benefits are accumulated by project numbers and the length of histories, including invisible values. But the graph of this page shows saving amount by pricing perspective. It is bigger and bigger by more project numbers, and it is reflected on the right graph. Of course, these savings get even better the more you use. As I mentioned in the first chapter, Ecolab regulatory team can support the resin substitution part, so you can consider the number of applicable projects in various directions. Ecolab promises you can get great support from every specialist of us. Thank you.
Thank you, Jake and Mats, for your insightful presentation. We'll now move on to the Q&A session. I invite our audience to submit their questions using the question box, and we'll address them one by one. So let's bring up the first question, which is directed to you, Jake. Is that between JA50 and AP plus 80, which resin is better to develop the actual process for production?
Oh, it is very brilliant questions. Okay. Um, the two resins have different characteristics, actually. Jetted A50 uses the high kinetics properties to induce sufficient DBC at a high flow rate. In order to use the high flow rate, about 10 to 13 centimeter is packed and used normally. This minimizes the consumption of resin. The increase in the cycle caused by less packing resin is compensated for by faster flow rate. On the other hand, AP plus 80 is a resin that can apply the most popular process condition. If you need to minimize the consumption of the resin, I can recommend jetted A50. And if you need to use the popular process condition, such as 20 centimeter packing height and 4 to 6 minutes residence time, I can recommend AP plus 80 to you. Thank you.
Thank you, Jake. So the next question is directed to Mats. So it is, what are the primary differences in developing a biosimilar versus the originator drug?
Well, that's a great question. I think major differences is that as you have the originator drug as a reference, you need to prove biosimilarity between the originator drug and the biosimilar drug by conducting a lot of analytical characterization experiments. When you have done that, you have sort of proven that you have a very equivalent molecule. And that means that you can actually reduce the amount of clinical trials required for the originator drug. And that's sort of the big difference. You can actually shorten the time to market from the 10 to 15 years for a new originator drug down to 6 to 8 years for a biosimilar drug. Of course, that also leads to much lower risk of failure in terms of clinical trials because you know what molecule you actually have. So that's sort of the major difference from a pharmaceutical company perspective.
Mats, could you also tell what are the differences between jetted and emulsified bead technology to our audience?
Good question. As said in the talk, Purelight, Ecolab have spent a lot of time developing the jetted technology, and that is now the standard for our all new resin NPIs, basically, all new resin developments that we conduct. The driver for this has been to sort of reduce the carbon footprint because you use much less solvents and water when you do jetting beads versus the classical emulsified beads. So it's a big saving there, sort of also in the context, of course, on sustainability and saving the planet. The other technical benefit is that you get a more uniform bead because you actually spray the beads through a membrane and you get almost exactly the same diameter for every bead. So it will be a very narrow particle size distribution as compared to the emulsified technology where you actually have a stirrer that controls the bead size and the resulting distribution of the beads. So that will you will get a much broader particle size distribution with emulsified. So, and a more narrow distribution also leads to better chromatographic performance when you are striving for high resolution processes. So that's another benefit. Could also ultimately lead to better packing performance because you have fewer fines, less risk of clogging the nets for a column, for example.
All right. Thank you, Mats. The next question is, what types of services are prepared for resin substitution? So I think this is directed to Jake. Could you please answer that?
Oh, this question is what I want to get from customer always, actually. Yeah. Regulatory support is available for the clinical phase from the preliminary. Documents are prepared for each phase of the necessary contents, and through meetings, a guide on necessary part is delivered and helped through professionals. Professional manpower consists of people who have experiences in actual resin substitution. Ecolab will guide you by announcing what kind of documents and experiment should be prepared detailedly. So please contact us. We can help you.
All right. Thank you, Jake. The next question is for Mats, specifically for biosimilar development and manufacturing. What are the benefits of Ecolab's jaded resin bead?
Indeed, interesting question. You know, an obvious benefit is that since we are using agarose technology, agarose chemistry, and the majority of the originator processes are based on agarose-based technologies, you have sort of a much smoother and less risky transition from the originator drug to our resin ultimately. You can sort of predict the outcome since the chemistries are very similar. It should also lead to less time to spend on the lab bench doing verification experiments and related activities. That's on the development side. If you also think about the commercial side of things, when you ultimately reach the commercial scale, security of supply is a key element in this industry. The commitment to deliver resins to the same quality over decades. And this is a commitment that we have in Ecolab, as said, building out capacities, working a lot on supply security and related topics. So those two elements, predictability in PD scale and security of supply at commercial scale, I would say, are the two main benefits.
Thank you, Mats. So, since we have limited time in hand, we'll just take one last question for Jake, which is several purolite resins were shared during the presentation, but it can be difficult to know the use case for each. So, could you just simplify the purpose of jetted A50, AP+80, HIP H, and CH1?
Oh, it is a good chance to introduce almost platforms we have. Thank you for having interesting about this. As you know, the description of jetted A50 and AP+80 has been made earlier. The last questions I had. So I tried to explain about DuraCycle first. Mm. It is durable to 0.5 normal to 1 normal sodium hydroxide. Therefore, it is preferred for target process development where protection and CIP and SIP for bioburden are required. As you know, the elution of protein A resin is usually done at the pH lower than pH 4. Sometimes there are targets whose yield is reduced by this condition. So high pH is prepared for acidic sensitive targets. Because of that, the naming of high pH is the region about that. So to summarize, the high pH is characterized by elution from pH 5. CH1 resin is ready for development of FEP and bispecific and is a non-protein affinity resin that can bind to CH1 site of the immunoglobulin. Okay, thank you. I hope that it is what I want to know. Mm.
Yeah. So thank you, Mats and Jake, for such insightful presentations and for answering the questions to our audience so comprehensively. So that we have reached the conclusion of our today's webinar. I'd like to extend our sincere gratitude to both our speakers and our audience for being such an attentive and engaged group. We hope you found today's session informative and valuable, and we look forward to welcoming you in our future webinars. Thank you and have a great day ahead.
Thank you.